record this for the final. You have your final study guide. Use it. Today we're going to go over one last thing. This week we're going to go over photosynthesis and cellular respiration. But to just bullet point it real quick, cell organelles, and function. You have to know what they are. What does each organelle do? Where are they? Cell types. You have to know which cell types are which and which has which organelles. You have three major ones, two big ones, prokaryote, and eukaryote. Prokaryote is a bacteria. Eukaryote, you have animal, plant, fungi, and protist. So you have to know each one of these has different structures. This one, no nucleus. Again, I'm just going to make a real quick summary by doing a Venn diagram with 4 circles. That looks a little complicated. What do you think? What if I do different colors? And I say, this is animal. Well, an animal cell has a nucleus. Golgi body, endoplasmic reticulum, rough and smooth, RER and SER, vacuoles, cell membrane, all that, right? All the cells, all the eukaryotic cells have those. Animal cells, mitochondria, let's not forget the mitochondria. All the animal cell, all the cell types have mitochondria. All the eukaryotes have mitochondria. What you would do best is to try to make this chart on your own. It would be the best thing you can do for yourself, is to make this chart on your own. Can you do this on your own? Do you know? You don't have to make a Venn diagram. You can make something simpler. But you should be able to identify what organelles go with which cell type. So if we would do a plant cell, plants have all of those. They all share all those in common. They share with fungi and some protists fungi and some protists, plants share cell wall. Plants have chloroplast, though, that fungi and protists, well, do some protists have chloroplast? I'd have to look that up. Protists are strange little organisms. But for sure, fungi don't have chloroplast. So now, if we go to fungi, we're talking about yeasts and mushrooms, right? Molds. They have cell walls as well. By the way, their plant cell walls are made of cellulose. And they have something else that other cell types don't have, is a central vacuole. Now, fungi have cell walls, but they don't have chloroplast. So they eat. They have all these other organelles, but they don't make their own food like plants do. They eat. They eat dead things. They're detrivores. Does that make sense? So, yes or no? Does that make sense? Okay. And then the last is protists. They're all over the place. Protists are all over the place. They have cell walls. They have both protists. I'm thinking. Yeah, I think they have a contractile vacuole, and they don't share it with plants. I showed you in an amoeba. You remember that? They just kept pumping water out of the cell. All right. I think if you know that, you'll be able to answer a lot of the complicated questions. It helps. I think it simplifies a little bit with extra color. But you see that most of the organelles, most of the cells share it. Now, to make sure you're clear that a virus is not a cell. Hence, most people, probably the majority of scientists, don't consider them alive, though it's an argument. And it's a bare majority. There's a lot of scientists out there that argue viruses are alive. They're not. They want people to accept them as the exception to the cell theory, because what cell theory say? What does cell theory say? Guys, if you're not working, you're not gonna. Oh my God. The nightmare. The cell theory. What is it? What is cell theory? It's basic. It's right out of the chapter reading, mentioned a bunch of times in class. What's cell theory? Stop trying to get out of work. Cell theory. Come on. That's it. The basic unit of life is a cell. Basic unit of life is a cell. That's right. Now, a virus, continue playing and trying to find ways out of getting work done, but it's not going to help you in the end. No offense, it's just not. So the virus is not a cell. A virus like COVID or like any other virus is made of some kind of protein coat. It's made of a protein. Have all kinds of different shapes. They're very tiny compared to other cells. They're very small. Inside, they have either DNA or they have RNA, single-stranded RNA or double-stranded DNA. Somebody asked, do we need to know DNA? Yes, of course you need to know what DNA is. Of course, we covered it, didn't we? So we covered it. You should know what DNA is. So, you know, at this point, at this juncture, you should know DNA is... Why is this doing this? We know that this DNA, you should know at this point that it's made of A that binds to T, G to C. There's a little bit of DNA inside a virus. The virus just gets taken up by the cell and copies are made, and that kills the cell generally or makes someone sick. And then this is RNA. And in RNA, you have A matches with U and G with C. And RNA has only it's a single strand. And you know that RNA is made of ribose, not deoxyribose, and that kind of thing. Are we all good with this so far? All right, so it's a good review. We're done with that. So what else should you know for a test? You should know how things transport across the membrane. What passes the membrane usually and what doesn't? What is the cell normally, without help, able or permeable to? What does that mean? That means, what is the cell allowed to go through or not? And that means the cell membrane. The cell membrane is structural. It's functional. It allows for both, it gives the cell some structure, also allows it to have some, it controls what goes in and out of the cell. So this is a very important structure. That's why we spend so much time with it. 2 fatty acids, a phosphate group, bilayer, not a single layer, not a micelle, but a bilayer, a fluid mosaic model of cell membrane. What normally goes through and what doesn't? Well, at this point, you should know sodium ion, any charged particle. I don't care if it's sodium ion or if it's phosphate with a PO4 negative. It doesn't matter. If it's negative, small or big, it needs help. It does not pass through easily, not permeable. It needs some kind of helper. At this point, you should know that water passes through somewhat, a little permeable to water, because water's so small, some of it gets through, very, very little, but does go in and out of the cell membrane. That's not new to anyone here, I hope. I hope that everyone here recognizes that sugar and proteins don't get through. They need help. None of these get through without help. What gets through? Anybody know what gets through? What is it that gets through the cell membrane easily? Small and nonpolar molecules. What are they? Oxygen gas, CO2 gas. They go through easily in both directions. I will complete. All right. So that's what gets through without any help. So how do you get stuff? How do you get these things through with help? What is help? Yeah. Some kind of transport protein. So again, looking at the phospholipid bilayer, looking at the fact that you have phosphate heads and you have these hydrophobic fatty acids. You should know this molecule called cholesterol. You see it all over the place. It's a ring structure, has multiple rings. I'm just sketching out a ring structure. It sits in the membrane. I did mention it before, but I didn't stress it, but now I am stressing it. So you should know cholesterol. So cholesterol is a lipid. All right. It sits in the membranes. It increases fluidity. It keeps the membrane fluid, increases fluidity. So it stops the cells from freezing. In the winter, you have more cholesterol in your membranes than you do in the summer. So it helps stabilize. In general, cholesterol stabilizes the membrane. Remember, it's always moving. And obviously, when it's hotter, it's going to be more fluid. You don't want the membrane to be too fluid. Then the other thing that you have, as someone mentioned a minute ago, is you have these things called transport proteins. There's several different kinds that we've talked about in class. The simplest of them being a channel protein that just allows something like sodium ion through going from high to low. Correct? That's the simplest of the proteins. There are other kinds of transport proteins we've talked about. There are some that require receptors and signals. They would have a channel as well, but instead of it just being an open channel, it has some type of receptor waiting for a signal. When that receptor binds to the signal, hydrogen bonding it's a signal molecule binds, hydrogen bonding changes this, and what that'll do is it will open the membrane, open the channel. So when that signal binds, starts off, the signal sent from somewhere else. What kind of things are signals? Hormones, neurotransmitters, right? So some kind of signal gets sent, and when that binds, it changes the hydrogen bonds change, and when the hydrogen bonds change, it changes its whole shape. When that shape changes, this opens up. Because the signal molecule attached to the receptor changes its shape. the door is now open. As long as that signal molecule, or what we call a ligand, right? Do you remember that word? Ligand. Once the ligand is bound to the receptor, the door opens and you can get whatever it is that's getting in, comes in. Again, moving from high to low. That's receptor mediated. We also talked about voltage channels. Do you remember that with the neuron? But you don't have to worry too much about those. The whole point is something opens a channel and things move from high to low. We call this, there's a word for this you should know because you're going to see it and probably see it on the test. Facilitated diffusion. Facilitated means what? To facilitate means to help. If I facilitate your education, I'm helping your education. If I facilitate your removal, I'm helping you being removed. So, help. And diffusion means what? High to low. So it's moving, helping things go from high to low, and here you're talking mostly proteins. Then there's simple diffusion. We talked about that, simple diffusion. Where do you see simple diffusion? That's oxygen and carbon dioxide. It's just going from high to low. No help necessary. Water does that too. When water diffuses, what do we call water diffusing? Osmosis. So all of these are examples of what we call passive transport. Meaning no extra effort necessary, no extra energy required. No extra energy required. This just happens, goes from high to low. All these are examples of going from high to low. High to low, high to low, high to low. There is no extra effort necessary. However, there are examples where you need to go the opposite direction. And we covered that. We talked about it. It was quick. I want to make sure I go over it again with you, because it's that important. I think it's important that we talk about it. There is something called active transport. We spent a lot of time talking about a battery. We talked about a cell membrane can act as a battery. We said a neuron does that. That's right. Uses ATP is correct. Excellent. It's going to use ATP, and in general, there's two things that tell you it's active transport. If you remember this, it probably gets you through every question. Number one, it uses energy. Usually ATP in a cell. And two, it goes from low to high. And the big always the example they use in every book is when you're going from high to low, no extra energy is required. But when you're going from low to high, you have to push that thing uphill, correct? So that's the example in the book. That's the example in every book. That's really the idea that you need extra energy to get it done. If you have this membrane and it's in a neuron, is it time to go? Is that why people are moving? Okay, good. So in a neuron, you have one side's more positive than the other. One side's more positive than the other. And you have these voltage-gated channels. Remember, we talked about them. There are ligand-gated channels. I just drew one earlier. And you have these channels that are voltage-gated. Depending, if you can make it more negative, this will pop open. But you have to change the volts, right? You have to change the ions. And that requires a ligand-gated channel neurotransmitter, allows ions to flow, which starts the domino effect, where you start to get a discharge of the battery. And what's a battery? The cell is a battery, because this side is more positive and this side's more negative. We call that a battery. Why is it more positive? Because there's more positive ions on this side. Maybe there's a ton of them over here and there's only one over here. When you discharge the membrane a little bit, this opens up, and what's going to happen? The ions are going to flow, correct? And as the ions flow, they go down into the membrane. That discharge, that shows up on a screen. You've seen this screen 100 times on TV shows, 1,000,000 times. It goes bleep, 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 correct? Right? And once it stops, it goes correct? So these are discharges of the membrane. Repolarization, these are depolarizing, meaning they're becoming less polar. They're discharging the battery just like your cell phone. Well, what has to happen to recharge the battery? So here, let me follow along, just like on the TV shows. It discharges, then you have to recharge the battery. Discharges, recharge the battery. So this is the discharging when the positives flow through from high to low. That's fine. This part's passive. It's ligand gated and voltage gated. Fine. But what happens? How are you gonna make it polarized again? What do you have to do? Nobody? Just guess. Nobody knows. It's not something any of you know. What are you gonna have to do? So now you got a few, let me draw it out for you. You have a few positives on this side. You have a few positives. They're equal now. You've depolarized it. Maybe even you got more positives down here than you hyperpolarized it, so you actually made it more positive inside than you really want. You have to have a helper. So here's your helper. Let me redraw this. You have to have a helper. When the helper is closed the way it is now, nothing's happening. What's this thing going to do? What do you have to do to make it polarize again? I'll say the question again. The inside right now is slightly more positive, correct? And the outside is a little more negative. We have to reverse that. How are we going to reverse it? Okay, so do I have an answer? All right, go. That's right. We're going to use ATP that you made during cellular respiration, and we're going to move potassium ions. Potassium ions, you see them here. You're going to exchange potassium ions on the outside. You're going to bring them in. And you're going to get 3 sodiums out. Now, each sodium is a +1. Each potassium is a +1. So 2 going in, 3 going out. what are you going to end up with? What's going to happen to the membrane? It's going to become more positive on the outside. And you're going to use ATP to do it. This is called the pump. Sodium-potassium exchange pump. You'll find this example and this explanation on this website. It's a nursing website. I just want you to see how important this is. This is in nursing and dentistry and medical school, any biologist, anybody intent on understanding any living cell needs to understand the sodium-potassium exchange pump. This explains muscle contractions and thoughts. So be ready to talk about this when we get back tomorrow. That's a classic example of active transport. So you see here, I put the website where I got this from. It is actually a really great website. It explains everything about the sodium-potassium pump. Where is this? What is the website? Is it a physicist website? Is it a research biologist website? Is it some nerd in a lab's website? Whose website is this? Nursing. Simple nursing. If you're going to be a nurse or a doctor, you have to understand cells. You have to understand the sodium-potassium exchange pump. Like, you have to understand that this is how it works. If you gave, there's a case that I remember, anecdotally, there's a case I remember, I don't know how true it is, but I remember reading about it, and it's a long time ago, so my memory might be faulty. But there was a nurse at the Cleveland Clinic who was given potassium to give to a heart patient. You need potassium to make your heart work, right? Because you need to bring potassium in and so to get sodium out to make the outside positive to recharge your cells, correct? Otherwise, the heart muscles stop working. So you need to keep that polarization so that you can depolarize it, discharge it, so you can make contractions, so you can pump your blood. Well, the nurse, for whatever reason was in her mind, took that potassium and instead of giving it orally, where it gets absorbed through the intestines slowly, she crushed it and put in the IV. So then, now all that potassium came rushing right into the blood. So this all became positive real quick, which made everything not work well. And the patient had a heart attack and died. So not understanding how these ions flow and which direction they need to go means you don't understand how a heart works, you don't understand how muscles work, you don't know how brain cells work, and that person you're supposed to be taking care of could end up dying. So when you're learning these things, you're not learning them just because. These are very important ideas. Osmosis leads to swelling. Anyone who's been on their period knows that at least, or even if you don't get it yourself, you know people that get swelling in their joints. They get swollen. They get what you call bloated during their premenstrual cycle, correct? At least you know some people that happens to. You get cramps. What are cramps? Contractions of muscles. Why is it happening? This. Ions are in the wrong side. Hormones are leading to excessive dehydration or excessive movement of fluids, which then leads to cramps. Athletes, anyone who's worked out has had cramps. We don't know all the reasons why people get cramps, but certainly it's this activity, the movement of ions, the concentration of ions on each side of the membrane leads to excessive contraction of muscles, which is what a cramp is. Everything in life is based on these simple ideas: osmosis, diffusion, active and passive transport. Are we clear on this? Okay, so now let's talk about synthesis. Did I just erase everything? This organelle. There's two organelles I want to talk about and you need to know before we move on. And these two organelles, they deal with energy. And when you're talking about the cell and you're talking about energy, mostly you're talking about ATP. You have to get ATP. But this doesn't happen in a vacuum. You don't get ATP on your own. Everything works in a system. I'm about to tell you the story of the endomembrane, of the origin of compartmentalization and something called endosymbiosis. Endo means what? What does endo mean? Inside. Endo means inside. These vocabulary words I keep going over with you will show up on the SAT. These prefixes and suffixes will show up on the SAT and the ACT. I can guarantee it. I have a book at home. I have 2 copies. I couldn't find the first one. Ms. Perez gave me the second one again from our last meeting. There's 300 words in that book that are important for anyone wanting to go to college. Guess what some of those are? The words I keep emphasizing on the screen. The prefixes and suffixes I keep telling you. Don't be surprised. Don't ask the question, how did so-and-so get such a great score on SAT, and why didn't I, if you're not working on these vocabulary words. You know the reason why. I'm telling you. Symbiosis, its root word is symbiosis, which means close relationship. So symbiosis is not good. A symbiote is not good or bad. A parasite, for instance, is a symbiotic relationship. So is commensalism. So all these are close relationships. There's not a good or bad about it. It just means close. So endosymbiosis is a relationship inside. So we have some observations. And I'm going to list to you the observations. There's 2 organelles. One is a mitochondria. And there's some interesting things about a mitochondria. It's one of the organelles you should know. And the mitochondria has 2 membranes. It's interesting. It doesn't just have one membrane. If you look at other organelles like endoplasmic reticulum and Golgi body, those are made of one membrane. The nucleus is made of a membrane. The mitochondria is made of 2 membranes. The mitochondria has its own DNA. Isn't that weird? All the DNA that you have inherited from mom and dad, all your DNA, and you built it in class, as you know, is linear, correct? It goes from 5' to 3'. It's anti-parallel. It's double-stranded. It's AT and GC, correct? Hydrogen bonded together. Everybody knows that. Should know it. The DNA in the mitochondria is circular. It's still double-stranded, but it's circular. Isn't that weird? It makes its own DNA. Its ribosomes, another organelle, are small. It has its own ribosomes, codes for its own ribosomes. Our ribosomes are big. That's all the rest of the ribosomes in your cell are big. The ones in the mitochondria are small. And the mitochondria have their own genes, and the mitochondria have two membranes. The mitochondria do their own division. They do binary fission. All on their own. They make themselves. Now, the other interesting organelle that's very similar also has two membranes. One is stacked like this. The other's on the outside of the organelle. Now, these are organelles inside your cells. Obviously, chloroplasts are not in your cells, but they're in plant cells. These are chloroplasts. They have two membranes. They have their own DNA. Their DNA is double-stranded, but it's circular. They have small ribosomes. They make copies of themselves. This is known as binary fission. Now, isn't that interesting? That these two chloroplasts have their own DNA. They do their own replication. Okay. Well, I'm going to give you a third, something you should know already based on the assignments you were given on Friday in your chapter reading. You know of these things called prokaryotes. Prokaryotes have one membrane. They don't have any internal membrane structures. But interestingly, they have double-stranded DNA, and guess what? It's circular. They only have one membrane. They have their own DNA. It's circular. They have, and by the way, they're small. They're all the same. So ribosomes are small. Same smallness as the chloroplasts and mitochondria. And they do binary fission. Huh. How do we explain this? How do we explain that these things that are living in every one of our cells have their own DNA? They do their own division. Where do we get our DNA? From our parents. So DNA is essentially what helps define us. So these things are different from us, are they not? But yet they're part of us. Huh. Now, what I just listed are facts. Now we're going to talk about our theory. And the theory makes sense and explains what's going on. Remember, theory has to explain what's going on. So the thought is that long ago, somewhere in the ocean or a lake, there were these single-celled organisms that developed. They were all at first some type of prokaryote. Nothing had a nucleus. Nothing had a compartment. At some point, this bigger cell, every one of these cells is doing something different. This little cell, I'm going to draw it in a different color just so that you can see what I'm talking about. This little prokaryote, it was small, compared to the bigger one. It could make and take apart sugar really quickly and make ATP very well. So it had ATP, a prokaryote that was good at making ATP. Now, when one cell eats another cell, I showed you this, we called it phagocytosis. Do you remember that? One cell, a big cell, I showed you an amoeba eating a bacteria. Some cell comes into and wants to eat this cell. It can't bring it in through a protein. No helper protein will help bring in a giant cell. Big things have to be taken in by something called phagocytosis. Cytosis, again, cyto means cell, phago means swallow, so the cell swallows the other cell. So the way it happens is that the cell starts to change shape. And this I could show you we see it happening all the time in nature. We could take a look at it underneath the microscope if you like. But what happens is, as the cell changes shape, whatever it's taking in gets swallowed up by the cell. Then the cell pinches off, and what happens is whatever is being swallowed finds itself inside a vacuole. These are all organelles that you should know. So this is a food vacuole. This membrane used to be part of this membrane. Does that make sense? This membrane used to be part of this membrane, but it pinched off. Now the bacteria is sitting in here. So you need to kill it, and you need to break it down into pieces. You need the protein, the lipid, the DNA, and the sugar. You don't need a bacteria inside your cell, correct? So you need a killer. You have these things called, there's two of them, lysosomes. Another one, a very similar one, is peroxisomes. Lysosomes have killer enzymes. They destroy anything that comes. These enzymes will break you down when you die. When you die, you'll be turned into a mush. You get turned into a mush because of these killer enzymes in this lysosome. So this lysosome binds to this. You don't want these enzymes floating free in your cell, because what are they going to do to you? They'll break you down. So you keep them in a vesicle. So when this membrane here fuses with the food vacuole, what happens to the lysosomes? They go into the food vacuole. What happens to the bacteria? It gets broken down. So now the food can go and become part of your cell, and you can eat. That's how you digest. That's how you break things down that are really big. Now the little pieces and parts can be used by the cell, and whatever you took in is not a danger. That's called phagocytosis. Is that clear? Does that make sense? No questions? Okay. So long ago, this big cell found this little cell. It's going to eat it like it always does. It surrounded it like it always does. But something weird happened. For some reason, this cell would not break down. For whatever reason, that stayed there. How many membranes do you see there? 2. One from the cell that tried to eat it, and one from the bacteria, right? 2 of them. Do you see 2? All right, 2 membranes. This was its own cell. So it has, what does it have inside it? No, this is a bacteria. What does it have inside it? DNA. Circular DNA. Well, over time, they developed, since the big cell couldn't eat it, if you can't beat them, join them. So they became partners. So now this big cell had a cell inside it that had a membrane and its own DNA. The big cell put sugar in. It went and got the sugar. It ate things. Nobody was going to eat him. He's big. He's eating things. He's bringing the sugar to the little cell. What's the little cell doing? Taking the sugar in and making what? A lot of ATP. And it gives ATP to the big cell. It uses some of it, but it makes a lot of ATP for the big cell, so the big cell's happy. The little cell's happy because it gets sugar without risking its life. Correct? And when it needs to copy, it makes copies of itself and goes on. This, over millions of years, became the mitochondria. There were other compartmentalization events, other events that created compartments. And eventually what you had is you had a cell that was a eukaryote that had all these membranes, that had ribosomes on it. This was the rough endoplasmic reticulum, the RER. There was a smooth endoplasmic reticulum, SER. There was the Golgi body. As you all know, there are vesicles. There were all kinds of food vacuoles and lysosomes and peroxisomes, all kinds of crazy little things. But there was this really interesting, and they made copies of themselves, so there's all kinds of copies of them. These little things called mitochondria. They were making ATP for everybody in the cell. This was a eukaryote. So all eukaryotes had what? Mitochondria. So time went on, as time does, and this cell found another cell. Now, it had mitochondria, that's true, but it still needed to get food. It was still eating. It still needed to get food. So how is it going to eat? How does a cell eat? It uses what process? If it's big things. Remember, this is millions of years ago. It's only single celled. No animals, no plants. This is a eukaryote, but it's not an animal. How does a cell eat? I just told you. Phagocytosis is correct. So it's going to use phagocytosis. It finds another cell. Here it is, tiny little thing. This guy can do photosynthesis. That means it can take light and water and CO2 gas. You can take all those together. We're going to look at photosynthesis in a little bit, tomorrow actually. Yes, it's on a test. And take and make sugar, make glucose. So it was really good at doing that. This cell couldn't do that. This cell has to eat. To get energy, to get sugar, this thing has to eat stuff that can make sugar. This guy can make sugar. So through phagocytosis, we know what happens. I just drew it all out for you, but I'll do it one time here. I'm not going to add all the organelles if you don't mind. The cell, the big cell, eats the little cell. How many membranes are you going to end up with? 2 membranes again. So here you end up with 2 membranes. This became what? Over millions of years became what? No, mitochondria is already made. These already have mitochondria. These cells already have mitochondria. They were yellow. Chloroplasts. These cells became chloroplasts. So all the cells that didn't happen to, they kept evolving. Eventually, some of them became yeast, and a group called what? Fungi, right? They are eukaryotes, right? Another group, another branch, they led to a group called animals. They have mitochondria. See that? Do they have chloroplasts? No chloroplasts, but they have mitochondria. This group went on and became what? Plants. The only ones with chloroplasts. The only ones with now they're not the only ones that can do photosynthesis, because way back here those bacteria did not go away. They're still here. Some of those bacteria we call cyanobacteria, they're still around. They do. Cyanobacteria are bacteria that do photosynthesis. They're still around today. In fact, when we compare the DNA of cyanobacteria to that of chloroplasts in the mitochondria, in the chloroplasts, the DNA in the chloroplasts, the DNA in cyanobacteria, they're very similar. This is a story of compartmentalization. How did we get a cell like ours? It's so complicated with so many different compartments and do so many different things. This is one way. Now, cyanobacteria are really the reason we're here. This is one of the key organisms in the history of planet Earth. Planet Earth used to be a very long time ago, when the continents were different continents. You know there was Pangea, yes? I figured you heard of those. A long, long time ago, the planet was not green. The planet was purple. Planet Earth was not green. If you look at planet Earth today, you see it's blue, right? Blue water. But there's a lot of green. Like the plants are all green, and they did. But our planet at one point was very purple. Isn't that weird? That's because there are these organisms, these little purple organisms, these little bacteria that do photosynthesis. In a weird way, they don't produce oxygen very well, but they do photosynthesis. They make sugar from light, and they make sugar. So they do that. But they don't really make oxygen, and they don't do the photosynthesis like we do it. But along came this other bacteria, a little green bacteria, that we call cyanobacteria, that could do photosynthesis really well. It outcompeted them. It didn't kill them directly. It just took up all the space, all the resources. It used a green pigment. Obviously, the purple bacteria used a purple pigment, right? These are pigments. This one used a green pigment. The green pigment, you might call a pigment artist. What would you call a pigment? What is a pigment? You're an artist. Okay, it's not a color is what we see. What's a pigment? I like that idea. That's a really beautiful idea. It actually turns out to be a shape, is correct. But it's a molecule that absorbs light. A pigment is a molecule. So let's go back and define what a pigment is. A pigment is a molecule that absorbs light. Cyanobacteria. That's the big one. Now, this molecule that absorbs light, if it absorbs light, what does that mean? It's taking the light in, correct? Are we going to see the light it takes in? No. What light do we see? Okay, let's take a look at skin color. My skin color, don't argue with me, is brown. I can't tell you how many kids argue with me about this. And I could pull out it is a shade of brown. Bunch of jerks. So it's a brown skin tone, albeit a lighter brown. And what you see here is the color, the light that is not being absorbed is what color? It's brown, which is a mixture of different frequencies of light. The darker you are, the less of this light is being released. True black is the absence of light, which means you've absorbed all the light. No one in this room is true black. We've made true black. True black is a pigment that we've made. It's actually phenomenal. You could look at it and it looks like it's a black hole. You don't see anything. Nothing. There's no light bounces off it. Now this book bag is the color purple because it absorbs all colors except for purple. So a pigment absorbs light, and the light that it doesn't absorb is the color you see. All right, the color, so in a green pigment, that's the light that's not being used. So this cyanobacteria took over, and the interesting thing about cyanobacteria, it was really good at taking CO2 out of the atmosphere and producing oxygen in excess. It produced a lot of oxygen gas. Before cyanobacteria, most of the earth was CO2. There wasn't a lot of oxygen. We know that because when we've dug holes really deep. One hole was dug so deep that the Russians hear sound. It's in Mongolia or somewhere. The Russians dug it. The sounds they hear, some people say that sounds from hell because they've dug the hole was so deep. But when we dig deep enough, we see that rocks are as old as these at the time of the when the planet was purple, when there was a lot of CO2, not a lot of oxygen. Do you think we find what do you think we find about the iron? What? What do you think's happening to the iron that we find that's a few hundred years old, a few million years old? What do you think about that iron? What do you think what form does it look? What does it look like? Rusty. What is rust? Another word for rust is oxidation. That means you're adding oxygen to it. So when you take iron and you add oxygen, you get iron oxide. You call that rust. Artists, oxide pigments are some of the most beautiful pigments. So iron oxide is the rusting of iron, adding oxygen to iron. So if this planet didn't have oxygen, when we dug deep enough, what do you think about that iron? Is it rusty? No, it's not. When we dig deep enough, we find iron that's not rusted. How could it not be rusted? There wasn't a lot of oxygen. There wasn't a lot of free oxygen. I mean, the oxygen was there. It was just bound to something else. Likely to what? To carbon. It was probably CO2. Well, this cyanobacteria, I'm going to spell it again so everybody gets it. Cyanobacteria changed the atmosphere of the planet. It made it so the atmosphere had oxygen in it, had less CO2, and led to the ability of life to flourish on planet Earth. It took 1 billion years or so because cyanobacteria is small, but eventually what you got was a planet Earth that obviously a lot of blue water, but also had a lot of green land, a lot of green plants. It's a lot of blues, a lot of greens when you look at planet Earth today, where in the days before life flourished on planet Earth, before cyanobacteria, it was all purple, a lot of CO2, not a lot of oxygen. We know that because we know we can look at the rocks and we know what happens when you put iron in the presence of oxygen. Anybody that has a car in Cleveland knows what happens. It rusts. So that's pigments. Cyanobacteria changed the world. This is going to be a key idea you might see on the test. We're going to talk about photosynthesis tomorrow. This is endosymbiosis. You need to know endosymbiosis. You need to know it. So don't get lazy. Make sure you know this by next week. You need to know the difference between prokaryotes and eukaryotes. You know the evidence for endosymbiosis, the fact that they share DNA. You need to know what the form of DNA is. You need to know what phagocytosis is. You need to know that mitochondria is in every eukaryote and that mitochondria makes ATP from sugar. You need to know photosynthesis makes glucose from carbon dioxide. And they ask very tricky questions that we're going to go through this week. So if you're paying attention, you should be able to do well on the test. But they ask very tricky questions. They don't come out and ask you, what does photosynthesis make? They give you an experiment and they tell you, if this is made, what's happening? If photosynthesis is happening quickly, what happens? If I take away this, will photosynthesis keep happening? If I add this, will photosynthesis keep happening? They ask very tricky questions. Yeah. It's 2 hours long. So depending on how many of those experiments I add to the questions, because I'm going to have it 2 parts. One's going to be on paper that you're going to answer in Schoology. Okay? It'll be in Schoology as well. And the other one, I'm going to give you a copy on paper. Well, maybe I won't. Maybe it'll just be all in Schoology. I'm not sure. But there'll be a part in Schoology, and there's going to be a part on the OST site, the Ohio State testing site. Is that clear? So you have two sets of tests. Are we all good? Yeah. You've all done the state testing with Mr. Pennypacker, correct? Yeah. All right. So, have you done state testing on the website before? Are we all good then? All right. I'm posting this 4th period.